Good afternoon, everyone, and welcome to the Pacific Salmon Commission's Seminar Series on Environmental Change and Pacific Salmon Management, the view from both sides of the border. It's a couple minutes after 2. Participants, looks like the numbers are still slowly going up, but cognizant of time, I think we will go ahead and get started. My name is Sarah Martin. I'm a stock assessment biologist with the Department of Fisheries and Oceans based out of Kamloops, and I am a member of the Coho Technical Committee. Today, I have the privilege of introducing our 27th seminar on the effects of capture and release and variability in hatchery wild encounters, featuring speakers Jason Seals and Dr. Scott Hinch. Jason Seals is the district fish biologist in the Mid-Columbia District for the Oregon Department of Fish and Wildlife. He and his team do their best to manage several high-profile salmon, steelhead, and native trout sport fisheries, primarily in the Deschutes and Hood River basins. They also manage population, life history, and creel monitoring programs that provide information directly to management of these and other regional sport fisheries. After 30 years of fish research, monitoring, and management, his favorite fish species, mostly because of nostalgia, is probably the Dolly Varden. Jason's presentation is titled Differences in Relative Catchability of Wild and Hatchery Origin Steelhead in the Deschutes River, Oregon. Dr. Hinch is a professor and associate dean in the Faculty of Forestry and Environmental Stewardship at the University of British Columbia and is the principal investigator of the Pacific Salmon Ecology and Conservation Laboratory. His areas of research include salmon migration, behavioral physiology and ecology, land use effects on fish ecology and habitat, and adult salmon migration survival, behavior, energetics, physiology, habitat use, environmental cues, effects of fisheries and capture release, passage and regulated rivers, disease and pathogens. The title of his talk today is Release Mortality in All Pacific Salmon Fisheries Along the Homing Migration. Before I pass things over to the presenters, just a reminder on the format of today's seminar. The speakers will present back to back, and then we will have a moderated Q&A session, so please hold questions until after the second presentation. And another reminder that every seminar is recorded and made publicly available on the PSC website and YouTube channel. So if you know folks who are interested in today's topic but were unable to join, the seminar will be uploaded and available online soon. And I think with that, I will turn things over to Jason.
Thanks, Sarah. Can you hear me okay?
Sound great.
Okay. Okay, as Sarah said, I'm Jason Seals, and I am the district fish biologist for the Oregon Department of Fish and Wildlife in the Mid-Columbia District, which the Deschutes River is a big part of our district. And I started here in the Deschutes over 20 years ago and started managing these projects and the data that's associated with it. And, one of the first things that I noticed was this issue, which is basically the difference. There was a very noticeable difference between the stock composition. And when I say stock composition, I mean hatchery versus wild. So I noticed the difference between catch in the sport fishery and catch in our monitoring locations in our traps. And the difference is that basically they're opposite. So I'm going to try to sort of tell that story here today, and hopefully you'll think it's nice and interesting. So I guess just to back up a little bit too, not, I mean, we noticed and tracked these differences for quite a few years, and then a couple of years ago we did publish this. So after this, if you're interested in really sort of doing a deep dive into, or the paper did more of a deep dive and into the nuances of this subject, but so I'm sort of don't have a long time to go down into the details of the paper, but this should give you a good idea. So I'm going to give you a little background on the Deschutes Basin, um, and give you a little introduction into the steelhead fishery. And like I described, talk about the differences in catch of hatchery and wild steelhead within the Deschutes sport fishery. And also look at the differences in catch of, looks like I have a typo there, between hatchery and wild steelhead within the adult monitoring stations. So this is map of basically half of the Deschutes Basin. Um, you'll see in the upper left of the map is outline of the state of Oregon. So the Deschutes Basin is in north central Oregon. It flows from south to north. So it meets the Columbia between the Dalles Dam and the John Day Dam. Um, I don't know if everybody can see my cursor here, but so this is the confluence area here. Um, as so these are summer steelhead, so they're entering the Columbia River starting in early July, and they're moving upstream over Bonneville Dam, and then eventually coming over the Dalles Dam and entering the Deschutes here. And this is really our study area. The area that's highlighted here is bold. And so this is about 40 miles from the mouth of the Deschutes upstream to Shears Falls, which is at river mile 43. So one thing that's important here is this is a mixed stock fishery. So um, basically every stock of steelhead that is going upstream through the Columbia for summer steelhead are not all of them are entering, but there's a lot of dip in and strays that pull up into the fishery. And so the fish that I'm talking about today that anglers are catching, it's a mix of all hatchery and wild stocks within the Deschutes, but also upstream of the Deschutes as well. So, and then also the John Day Dam. So with this study area, basically we were monitoring the fishery here in the lower 40 miles. And then we're also looking at the stock composition at the Dalles Dam, John Day Dam, and here at the Shears Falls Trap. So we have sort of the fishery surrounded with fish monitoring sites. So a little bit about the fishery. So these are summer steelhead, as I mentioned. So they're coming in into the lower Columbia in early July, and they start entering the Deschutes in about mid-July. And so the fishery really ramps up in mid-July, and it peaks um, generally in the first couple weeks of September, along with, the peak of the run. And then um, as September progresses and October, you get into October, fish start to spread out a little more, and the fishery really fades and spreads to other places. So summer and early fall is the primary effort. It is very hot and dry. It's basically the Deschutes is like fishing in an oasis. Base summer flows in the Deschutes are around 4,000 CFS. And that's primarily because water um, from the Cascades, filtrates down into lava flows, and then about halfway through the Deschutes Basin, there's a bunch of large springs that make it a very stable river, and so it's very conducive for, high summer flows, even though it is in a desert environment. Generally, we get about 10 inches or less of moisture annually. It is also very hot in the middle of the fishery. Generally temperatures range, high temperatures during the day range from 90 to 100 degrees, but water temperatures are generally around 60 degrees. As I mentioned, also, it is a mixed stock fishery with a lot of stray steelhead. So it's a lot of stray hatchery steelhead, but also a lot of stray wild steelhead from various rivers upstream of the Deschutes and the Columbia. Hatchery steelhead are adipose mark for allowable harvest. But as you'll see in the coming slides, wild steelhead do make up the bulk of the catch. Anglers come from all over the world to fish here, so it is very much a destination fishery. It is flies and lures only, so no bait is allowed. And one thing that is unique to the Deschutes is that anglers must fish from the bank only, so no fishing from a floating device. So they can use a boat to go downstream, or if you're in a jet boat, you can go upstream, but you have to leave the boat and be on the bank to be fishing legally. And spay fishing is probably currently the most popular method, although there is a very strong contingent of gear fishermen as well. So as far as the creel, we have been doing creel, so angler surveys since 1977. Um, and we have two creel locations. One is at Heritage Landing, which is right where near the confluence of the Deschutes and the Columbia. And then on the upstream end, we have the Max Canyon Creel Station. And so with these two creels, we're getting both ends of both the upstream and the downstream end of the fishery. We're not interviewing every access point, but it does get the bulk of anglers. And so on the Max Canyon Road, it is getting day users going up and down the Max Canyon Road. And then at the Heritage Landing site, there is a roadless section, so it getting boaters coming down the roadless section or jet boats that are going up and coming back down to Heritage Landing. And the Shears Falls trap is here, and I'll get to that later, but basically with the catchability study, we looked at stock composition in the fishery, and then at the Shears trap with the assumption that the Shears trap was the best representative of stock composition because the steelhead that are coming through the fishery are then also ascending the Shears trap. Um, so just looking at my notes here. So yes, we wanted to compare for the catchability study, compare the stock composition within the fishery and at the Shears trap, but also use the Dalles Dam and the John Day Dam as a control for stock composition and compare that to what it looks like at the Shears trap. So this is just I put in the number of angler days to give you an idea of the volume of the fishery. So these are the number of anglers that we're estimating annually, which is from July to October. So it's not the entire year, but it's during the bulk of the when people are fishing. So for most of the period on record, we were seeing from 8,000 to 16,000 anglers per year. And then recently, which is mostly driven by um, poor returns of steelhead, we have seen in the 2,000 to 8,000 anglers per year. So this is just an illustration of the angler catch composition from the creel. And um, as you'll see in this time series, and this is the time series that we used in the catchability study, um, for almost nearly all of these years, so this is just the number of steelhead by stock, wild or hatchery, that were observed by our creelers annually. So on most years, anglers were reporting, 2 to 3 times the number of wild steelhead as opposed to hatchery steelhead. So the only year where catch was where there were more hatchery. So keep your eye on 2006. That was the only year where there were more hatchery steelhead reported in the creel than wild steelhead, but it was pretty much equal at 5,400, so 5,400 hatchery fish and 5,300 wild fish. So as far as our fishery independent sites where we're doing population monitoring. This is the shears trap is what we call it, and it's a ladder steep pass trap. And if you can see here, this is the ladder that is ascending shears. This is Shears Falls here in this picture in the middle, and fish ascend this ladder, and at the top of the ladder is the trap, which is here in this top picture. And so it's a real-time trap in that fish ascend the ladder, and then we have water running through a steep pass, and fish swim up the steep pass and drop into a sampling tank, and the trap operator is waiting for fish to actually swim through the trap, and they're sampled and then passed above. Typically, we sample about 1,500 to 4,000 steelhead per year. And our sampling, as I mentioned earlier, is concurrent with the fishery monitoring. So while fish are swimming through the fishery, after they swim through the fishery, they are ascending Shears Falls, and we're sampling a portion of those fish that are ascending the falls. And unrelated to this, but the reason we do this project is we are doing mark recapture estimates for not only steelhead but also false Chinook and coho. So this is our observed stock composition at the Shears Trap, and as you'll see, the hatchery fish are in green again, and wild fish are in orange. And it's just the opposite of what we see in the fishery, where we're seeing, 2 to 4 times as many hatchery fish in our sample at the Shears Trap as we are seeing in wild fish. And again, here's 2006, where we saw, we sampled 2,653 hatchery fish and only 574 wild fish. And this is just our escapement estimate, just to give you an idea of the magnitude of fish that we're estimating annually. Um, here's 2006 where we estimated 25,000, almost 26,000 hatchery fish and only 4,000 wild fish passing over shears. So again, similar to our the stock composition where we're seeing 2 to 4 times as many hatchery fish as wild fish. So this was the just sort of in the catchability study itself, which was we published a couple of years ago, was basically looking at these differences in stock composition that I was just showing you, and we did a little deeper dive into the nuances of of this issue. And again, we wanted to look at the stock composition at the Shears Trap, the Dalles Dam as a control, and the John Day Dam as a control, and look at the stock composition within the fishery. And then we wanted to calculate probabilities of capture at each location. So this is just the illustration of the three different fishery independent sites. So where we were able to, um, you know, look at fishery monitoring sites and determine uh, composition. And so this is on the far left here is the Dalles Dam, in the middle is John Day Dam, and then on the right is the Shears Trap. And it's very similar with all three of these panels where the number of wild fish is dramatically lower in proportion to the number of hatchery fish. And the sort of cycle here and the difference per year is very similar, which with all three of these. And then we also, this is just sort of combining the data where we look at the proportion of wild by location for both the dams and the traps, just to see if the proportion of wild is similar among the tra between the traps and the dam sites, and it tracks through time where um, the proportions are similar and there's not a big difference between the trap site and the dam sites. And then in contrast, the both the dam and the trap sites, the proportion of wild composition was, 20 to 40%, whereas in the fishery, again, it was in the 60 to 80% range. So the proportion of wilds is much higher in the fishery than it is in all three of the fishery independent sites. And again, this is just showing combining that data where we have the angler catch in orange here, and this is the proportion of wilds, so the percent of wild steelhead in the creel, and then this is the proportion of the percent of wild fish at the Shears Trap. And so you can see again, this is just sort of reiterating the point of the difference between the two. So at the dams and the trap sites, we averaged 27 to 31% wild fish. And then at within the fishery, we averaged about 65% within the fishery. So in the catchability study, we did a series of mixed effect models, which I'm not going to even try to explain here, but what we wanted to do was we wanted to um, within each of our sites, so each dam, the both dams and the Shears Trap and within the fishery, we wanted to calculate the probability of capture between each location. And this was basically our conclusion was that in the Deschutes, wild steelhead were over 2 times more likely to be captured by a sport angler in the fishery than they were at the independent monitoring sites, despite the much higher proportion of hatchery fish within the fishery. So I guess based on these results, um, we're curious because we don't really know what are the mechanisms that are causing hatchery steelhead to have lower catchability. Um, And we should strive to have hatchery steelhead that have near or equal um, catchability, because I mean that's obviously takes pressure off of wild stocks within these mixed stock fisheries. But also it's beneficial for anglers to have hatchery fish that are more susceptible to angling. Um, and where catchability is higher for wild steelhead, obviously as managers, we should be cautious within these fisheries where there are mixed stocks to not have adverse effects from catch and release angling that might have population impacts. And that's what I have. And special shout out to my co-authors here, Michelle Jones and Ian Tatum and Jeremy Henderson, and support by ODFW. And all of our funding for this comes from Sport Fish Restoration, so U.S. Fish and Wildlife Service, and also our co-managers, Confederated Tribes of the Warm Springs. So thank you.
Thank you, Jason, for a really fascinating talk. I'm sure it's going to generate a lot of interesting questions. So I just want folks to hold on to those, or better yet, to click on the Q&A box at the bottom and start inputting your questions for Jason. We'll transition to a moderated Q&A after the next talk, but for now, we're going to turn it over to Dr. Hinch for his presentation on the effects of capture and release.
Well, appreciate the invitation to present some research from my lab. I've kind of changed the title a little bit to be a little more in line with the theme for today. So I'm just calling this Release Mortality in Pacific Salmon Fisheries. This is a really important issue because across all fisheries, whether they be recreational, commercial, large numbers of fish are released for a variety of reasons. Some cases up to 50% of the catch may have to be released, either mandated through regulation or in recreational fisheries for other reasons. So I'm going to be focusing primarily on the recreational fishery, although I'll bring in some of the commercial fishery work at the very end here. So one of the ways that we've been assessing post-release mortality over the last 6 to 8 years is with acoustic telemetry. This is where we would be capturing, tagging, releasing, and tracking fish, primarily Chinook and coho in different locales in BC. The map you're seeing there shows all those little dots are underwater seabed mounted acoustic receivers. They're managed by different agencies and units and groups. On the margin, you can see the legend. And I'm going to be focusing a lot on what's going on in A, and then I'll come back to what's going on in C shortly. So we have this broad scale acoustic receiver network, which is kind of the backbone of our field work. And on the left side, you can see us attaching an acoustic tag externally, which is where all of the tags were placed on fish. Our study approaches included a variety of standard angling methods and gear types. So we looked at hook sizes, attractors, terminal tackle, landing nets, how you release fish, air exposure durations. We collected data then on the fish themselves, looking at injuries to the fins and eyes, scale loss, bleeding. We took tissue samples to get stock ID. We looked at physiological condition and infectious agents through some genomic approaches. I won't be talking much about that today. And we also looked at water temperature as an overriding factor. So we varied the fishing experience to impose realistic stressors on these fish. So generally speaking, here's one of my grad students, Steve, catching fish. So the fish could either end up in a net, maybe on the bottom of the boat, and then it would end up in our sampling trough, or it could go straight into the sampling trough, or it might be never enter the boat. All the work is done in the water over the side of the boat. With all these different approaches, the fish eventually would be tagged and released. Just to show you some of the responses of fish that we're measuring. So we looked at things like bleeding. So here's a range of bleeding scores from minor to severe. You can kind of see what the minor and moderate looks like. Severe, in this case, this is a treble hook that got lodged deep into the mouth of the fish, and often your holding water turns into orange Kool-Aid. So it's a sort of range of bleeding levels we scored. We looked at eye injuries ranging from very minor to moderate to severe, where the eye has been removed. Often the eye injuries happen because the hook, when it gets bitten by the fish, pierces the ocular cavity from inside the mouth and then pushes into the eye from the inside. Scale loss turns out to be a really important variable. We categorized it by minor to severe. You can kind of see how we categorize that. We'll talk a lot more about this shortly. We looked at the fraying of fins. So here's what I would call a minor fray. Here's a more moderate one where the fin is split at least 50% more down the rays. And then we'd have severe injuries where it was split right to the base of the fin. So we're going to focus first on Chinook. A lot of our Chinook tagging work occurred in the Discovery Island region here in green. I'm going to be showing you figures that are all going to look similar. These are going to be survival figures to assess survival. We knew the stock origin of all of these fish from the DNA, and so if we knew they were supposed to be crossing one of these lines and they didn't, then that could be assumed a mortality. We also were interested in using this time to event analysis, which gives us a really good indication of when fish are disappearing from our analysis. So the last known detection is the event that you're going to be seeing. So we had our fish categorized into 4 groups. The good condition group, these are kind of our baseline or control fish. These were fish that had no air exposure. Pretty much they're perfectly looking, maybe a little bit of bleeding, but otherwise they were in really good condition. And then we had 3 different injury categories: limited, severe, and eye injury. Basically, as you go down this category, the injuries become, as the name suggests, worse and worse, from moderate issues to more severe issues. And we kept eye injuries separate, and you'll see why in a few moments. And so here's a type of figure you're going to be seeing more of these. These are the survival analyses figures. So we've got the fish categorized by their fisheries effects groups here on the right. And you're going to see these lines appearing on this proportion detected figure, these Kaplan-Meier curves. First thing to note is that a lot of fish died, or several fish died right upon handling before we were able to release them. So we called those dead on day 0. And so there's a huge difference. Obviously, the control fish, there was almost no mortality for the first 10 days. What we saw immediate mortality in several of the fish groups, all of them, in fact, with the most severe ones being the severe injury and the eye injury groups. Before I discuss the figure, I just want you to note that the dark lines are kind of your average response, and then you've got confidence intervals shown with the light dots. So you can see the confidence intervals for our good fish here in blue extending out. So they're quite wide here after 10 days. Could range anywhere from 80% to like 95% survival, depending on that confidence interval. What's really important is the difference between the control fish and the other group. So you really want to think about how the blue lines relatively differ from the green, yellow, and red lines. So one of the big takeaways is that very little mortality in the control group up to about day 14. And then we started seeing some issues with the control fish. But at the same time this was happening, we had declines, in some cases much more rapid declines in survival for limited and severe injury. The big issues here were at the severe injured fish, where we see about 20% higher mortality than the control fish at any point in time. And in particular, the eye injured fish, these are the red ones. Eye injury fish just kept dying over time, whereas the control fish kind of leveled off. And so we see like another additional 25% mortality by about 2 months in those eye injured fish relative to control fish. We wanted to look more carefully at mechanisms and in a shorter time scale. So we went to the lab, in this case, the Banfield Marine Sciences Lab on Barkley Sound. We worked really closely with the Sport Fishing Institute to help set this up and run it. So we were catching fish here, bringing them back to these tanks at Banfield Lab. There's the lab in the background. The holding study methods basically were to capture the fish, then measure, do things to them, transport them, sample them, and that took about 4 days to load up our tanks, and then we'd hold them for about 10 days, and we'd observe survival and other aspects of their injury progression. At the end of that 10 to 14 day period, then we would sacrifice the fish and sample them. I won't be talking much about this. We'll focus mostly on this period. So again, here you're looking at these same sort of Kaplan-Meier curves. Again, just look at the thick lines and the different relative differences over the 2-week period. If fish exhibited no bleeding, they survived much better than the fish that exhibited some level of bleeding. So some level of bleeding we saw in a 30, maybe 30% mortality, but in difference from the no bleed, that's about a 15 to 20% difference. So just to remind you, this is what minor bleeding looks like. This is what moderate to severe would look like. Scale loss, however, was a very dramatic result. So just to remind you, here's what a minor scale loss would be. Here's something that's more moderate to severe. And so no scale loss, again, minor mortality. However, when you have severe scale loss of 2 to 3, you can see that we have about 75% mortality. And that nothing really happens until day 3 or so, and then the wheels fall off the bus. And this nothing happening until day 3 is very typical response we saw not just with this species, we saw it with coho, which I'll show shortly. We've also seen it with other studies we've done in freshwater with sockeye. And so one of the big findings that we had is you need longer than 24 to 48 hours really to see a lot of these effects and to understand the mechanisms. But clearly scale loss is an important mechanism. Size is also important. The red and pink are large fish. There's almost no mortality in these large fish. However, the smaller fish, we start to see after day 3 or so, mortality ramping up. And what's troubling here is that these smaller fish, where the highest amounts of mortality are, these are the fish that you have to release, either through regulation or possibly they are legal in some cases, but they're not as big as you'd like, so you might release them. And now these small fish have a much higher likelihood of perishing after release. Large fish, on the other hand, probably going to survive your release, especially if they're in good shape. So overall, when we compared sort of the fisheries effect groups like we did with our telemetry study, the takeaway is that the fish with no injuries survive really well. And then as you start layering on different levels of injuries, by the time we get to that 10-day mark, you see the severe and eye injury categories certainly coming out as having the highest mortality. In this case, the difference between the control fish or the no injury fish and the most severe injury fish is about 30% or so, which is a really similar result in scale and magnitude to what we found in our telemetry study, certainly for the first 10 to 12 days. We also looked at different hook types, treble hook versus single hook. We found the treble hooks certainly caused more severe injuries, and in particular on smaller fish, they were causing higher mortality. So here we are again with predicted line and confidence intervals. So these lines aren't overlapping until we get down to about 50 centimeters and smaller fish. And so these smaller fish are very susceptible to mortality attributed to trying to remove treble hooks. Let's turn our attention now to coho. Same sort of acoustic telemetry design, except we're moving now down more into the southern portion of British Columbia, and we're focusing a lot in Juan de Fuca Strait. Most of our tagging took place just near Port Renfrew. We were tagging coho that we knew were heading eastward, and DNA would confirm that for us. And we're focusing a lot of our attention on to cross this Juan de Fuca receiver array, which takes about 3 days. So in this case, we categorize fish like we did with Chinook, a little different, but very similar still, into good condition, moderate condition, and poor condition fish. What we found is that the mortality to get to that line in about 3 days, about 17% for the good condition fish, about double that for the moderate condition fish, and even higher mortality for the poor condition fish. Now, I'll come back to talk about some of the mechanisms here in the middle, but this certainly seems a bit higher than we recorded for Chinook under similar time trajectories. But again, I'll circle back in a moment to that. We also looked at fish heading to Puget Sound, and that's a bit longer. It's like a 3-time longer migration to get there. And what we see is mortality as much higher to get to that line. So over twice the level of mortality in the good condition fish. And in moderate and poor condition fish, really high levels of mortality, almost twice as high as we see in good condition fish. So it does seem like coho might have much higher post-release mortality than Chinook, all else being equal. But as I mentioned, there is something else going on here that we need to talk about. And so we wanted to investigate this seemingly higher aspects of mortality in coho salmon. And again, we went back to Bamfield and did another holding study, very similar approaches to what we did with Chinook. In this case, we're going to be comparing net types and a particular contrast with hook types. And so I just want to show you something. Here's a typical fish-friendly net that we used. We used those for Chinook as well. Look at the behavior of the coho as it comes into the net. Now, if any of you fish for coho in the marine environment, you'll probably notice this rolling behavior. It's probably some sort of anti-predator behavior, but the fish rolls multiple times when it gets into the net, which really creates a lot more injuries for itself when it's in that net. We wanted to have another sampling approach that allowed us to get the fish in without touching a net. And so we used a sling. Now, the sling is not something that anglers are going to be using, but it's something we use to try to corral the fish in a really non-invasive way. But I want you to see that fish rolling again. It comes into the sling and it starts to roll. So this anti-predator odd rolling behavior tactic is something that seems to be more coho than Chinook specific, and it does have a role in outcomes of catch and release. Well, and again, I'll come back to that in a moment. So we also studied hook types, a single hook versus a tandem hook setup. So back to this rolling behavior. What happens when those coho start to roll in a net or just roll as they get close to the boat is they start creating these line wraps injuries on their bodies. You can see on this one, you can see it here, and you can see here like it rolled 3 different times around the net, around the line. And so the line itself is causing the injury as much as anything else. So we wanted to try to look at this without that line wrap phenomenon. So we developed something we call the ning, which is a cross between a sling and a net. And so what the ning is the same material as a sling on the bottom, but it's got more meshing material on the sides, and there's vents for the water to go out. So it's easy to lift out of the water, and when the fish gets in there, it's not rolling as much, and also it comes out without any marks caused by a net. We've already been approached by dozens of lodges to make these for them, so if you're interested, please reach out to me after. Okay, so with coho, we're going to be talking about dermal wounding, and so the same sort of curves here as we saw before. And dermal wounding, it would be either the fish was caught in a net, maybe it hit the deck because sometimes fish are slippery and they hit the deck, or it got line wrapped as it came in. So any of those things created dermal wounds. And what you see is if there was no dermal wounding, coho had perfect survival over that 2-week period, whereas any level of dermal wounding led to about a 25% mortality. Let's look at fins now. You can see these fins here are damaged. If you look closely, you'll see some bleeding occurring where the damage occurs. And bleeding are obviously points of entrance for pathogens, as well as it also affects their ability to osmoregulate when you start having cuts like this. What causes that is nets. So even fish-friendly nets are some of the worst offenders, and you can see why here. Here's a fish-friendly net, but as soon as you put a fish into it, the fins start to bifurcate as the weight of the fish pushes against the meshing material. And you can see this happening all through the whole part of the caudal peduncle there. So if we start looking at survival in relation to number of damaged fins, if no fins were damaged because of how we captured them or landed them, they had perfect survival nearly. However, when you have over half of the fins damaged, 4 to 7, again, you see this large drop off of around now 70% survival or 30% mortality. So a large difference, again, being affected, caused by fin damage. Finally, we're going to be talking about those hooks, and the big difference between a tandem hook and a single hook really is in how severe the injury is. So severe injuries down here in the bottom, dark gray, were about twice as high in tandem hooked fish than in single hook fish. And these are what sort of the severe injuries look like. Minor injuries, a lot fewer minor injuries with the tandem, mostly because they were expressing more severe injuries. The other thing about tandem hooks is they're really hard to get out of the fish's mouth. So we developed a difficulty index for extracting the hook, and the hooks were about 5 times harder to extract from fish with a tandem hook setup than a single hook setup. And you can see here on the top right, a fish being released with a single hook using a gaff release. It's really easy to pop the hook out, whereas you could not do that easily if you had 2 hooks stuck into the fish's mouth. What we also notice is oftentimes one of the two hooks would be hooked really deeply, and even if they weren't, and you used a net, one of the hooks might tangle itself in the net, which increases the amount of time you're handling the fish, and then you start getting scale loss. So let's just go back to this figure I started with on coho. And you may recall I said, we had good condition, moderate condition, poor condition fish going from the tagging area to the Juan de Fuca straight line here. The big thing here is all fish in this study had some amount of scale loss because we all used nets. We didn't realize the severity of these net injuries, and so we used standard fish-friendly nets. So even in good condition fish, there was some scale loss. So when you look at the comparison between this telemetry study and the holding study where we had almost perfect survival for fish that we didn't use nets and didn't have scale loss, we start to see a pattern emerging. And that is that, these good condition fish, had they not had scale loss, probably would have had much lower mortality. And similarly, these other mortality levels are probably elevated simply because of the use of those nets. So the implications of this is that any interaction with the dermal layer that creates an injury, a loss of scales, or a puncture of some form is going to reduce the chances of survival of a released fish. The good news is that only small changes to angler behavior and gear can greatly increase survival. So we came up with 15 best practice recommendations to minimize release mortality for marine Chinook and coho. We categorized them here into five groups. The big one was gear. So avoid large hooks, treble hooks, multi-hooks. Didn't talk about avoiding inline flashers. When we experimented with them, we studied the metabolic recovery of fish. It took fish a lot longer to recover from being captured with a flasher because they're fighting much higher drag. And so the longer to recover means you have to hold on to the fish longer, or there's putting the fish at greater predation risk when you release it. So we recommend not using inline flashers. Landing approaches, land them quickly, obviously minimize air, and the big one, avoid using landing nets, even fish-friendly ones, if catch and release is a potential option in your fishing day. Avoid touching them with your hands because the scales in marine environment fly off really easily, whether it's in your hands or on the boat or in the net. Anything you can do to limit the touching is going to benefit them. And part of that is releasing the fish at the water line, not bringing them on board the boat, like I showed you with a gaff or a tool. And the other thing is limit revival. You should only be trying to revive fish if they are to the point where they cannot swim at all. They're upside down, and you're basically reviving them to protect them. But if they're able to swim, you shouldn't be touching them. You should just let them go immediately over the side of the boat without bringing them on board. And the big thing that we found in terms of our size effects is that small fish have high mortality when released. In some cases, it's impossible to get a small fish to survive a catch and release event. So avoid areas where you're encountering small fish and stop fishing when you get to your quota, because even if you are continuing to fish for small fish after your quota, those small fish aren't going to do well when you release them. And there's no use risking fate because even larger, modest sized fish don't always do well depending on how they're captured and released. So stopping fishing after your quota is attained is probably a really good practice. We rolled all this up into this document, so if anybody's interested, they can find all the research results, at least up to a couple of years ago, in this document. There's other online educational resources. The Sport Fishing Institute of BC has come up with really great short videos, and these links are live. You can find them here. This is the YouTube version. Or if you're an angler, you must have the Fishing BC app because that's where your license goes. And on your app now, we have best handling practices as a short video and some short information provided there. And if you want to have more details, the Pacific Salmon Foundation has some of our materials on their website there as well. So just as I'm wrapping up here, I just want to put some of this in context for all of the fisheries. So we recently published a review paper looking at release mortality in Pacific salmon fisheries along the homing migration. And so we were looking at all types of fisheries and different locales, different species. This is also publicly available if you're interested at this site. So what we did is we obtained data from 30 studies on Pacific salmon catch and release fisheries or bycatch and release fisheries. We looked at coastal, estuarine, river, commercial, recreational. We examined intrinsic, extrinsic factors, just like we did with the rec fishery one, but we did this with all fisheries types. And then we evaluated risk based on how high release mortality levels were compared to the control or baseline states. So to put this in context then, risk was high when we had 26 to 45% observed mortality, and this was across all fisheries. Even recreational fisheries had this sorts of level of observed mortality. So the risk can be high for all fisheries, depending on whether the fish are bleeding, had high levels of scale loss, fin or eye damage, or low oxygen caused by crowding or exhaustion. So you can pick your fishery, you can pick your species, and you can have high release mortality if these criteria are met. We did find that air exposure and handling duration led to high mortality and high risk when water temperatures exceeded 18. This is mostly a freshwater issue. However, there were some instances in our marine recreational fishing when days were well above 18, and we did see a temperature effect then. Perhaps the highest risk environment was the estuary and lower river, because that's where we're seeing the most rapid physiological changes in these fish in terms of their osmotic abilities, their maturation, and the temperature changes. And so we did see higher release mortality in these environments. In an absolute sense, the highest mortality and the highest risk, therefore, was associated with commercial gillnet and purse seine fisheries. However, there are ways to bring down that mortality. So we did come up with some recommended best practices. I think one of the first things we learned, which isn't so much a best practice for fishing, but it's a best practice for scientists, and that these short-term observations were really poor predictors of long-term patterns. So management agencies that did 24 to 48-hour studies really didn't do us any services, because that information that eventually found its way into escapement models and other management models was flawed. So you really need to have at least a 10-day period to be able to observe and understand mortality. And ideally, you're hoping to tie that in with telemetry. Most fisheries can reduce mortality levels if they modify the gear and they modify their handling modifications. And so we did list a whole bunch of recommendations for modifying our current fishing practices, depending on the gear and locations, all aimed at minimizing stress, injury, and bycatch. And I'll just give you a couple quick examples here. So for example, purse seine fisheries that we studied extensively, one way to increase survival and lower the risk is by keeping the nets quite loose, not tight like this when they come to the boats. Keep them away from the boats, because the tighter the nets, the closer the boat, the more injury these fish were getting, the more scale loss was happening. And brailing in small numbers as opposed to big numbers, also, again, reducing scale loss and asphyxiation issues. Minimizing the time a fish is on the boat. We found that less than 4 minutes really helped increase survival rates of released fish. Close 2 minutes is your ideal if you can get it to that. And releasing fish with a chute rather than grabbing them or kicking them overboard. All of these things that minimize touching and move them quickly through the processing does help. And lastly, I'll just give you an example from freshwater. Here's a beach seine set. It's commercial indigenous beach seine fishery, goes on the Lower Fraser. We found that if you can use shorter nets, in other words, you're not going to catch as many fish, you're going to be able to release the bycatch fish quicker. You're going to be able to find them quicker. And also by not bringing the net onto the shore, as is happening here, keeping them in deeper water, you're going to be able to find those fish before asphyxiation and injury harms them. And so there are small things that can be done in all fisheries to improve survival outcomes. And so with that, I'd like to thank so many funders and collaborators, but I really particularly thank my grad students and postdocs on this list. And I also want to thank the Sports Fishing Institute of BC and BC Shrift for helping fund all this. Thank you.
Thank you, Scott. What another fascinating talk. Sure to generate a lot of questions. So just a time check, we're at 2:57. So let's just take 3 minutes. And we'll begin the moderated Q&A promptly at 3. I encourage people that have questions to put them in the Q&A box. So we are unable to see if your hand is raised. You're muted from the presentation. So please put your questions in the Q&A, and we'll get to them at 3. So stay tuned. Thanks. All right, so we have some questions that are starting to come in, and we have both our speakers available to answer. So the first question, do you want to pose that question, Gary? one of our panelists?
Sure, thank you. Quite interesting in terms of the research, Scott. Did you have any way of estimating the probability of the severity of injury? Because, if your mortality rates are dependent upon the condition, and for fisheries analysis or modeling efforts, we have to be able to figure out what proportion of the encounters are going to end up in particular conditions.
We did odds analyses to look at what the increased odds were of various combinations of injuries resulting in mortality, and those are published in those papers I mentioned. Those Kaplan-Meier curves are more integrative than the odds, because they're not trying to examine necessarily individual factors when we start looking at all fisheries condition components. But in terms of whether probability of a fish getting these injuries, I think it's safe to say that since we set this stuff up experimentally, that if you were to come on the board with a fish in this particular situation, we will give you the odds of the likelihood of you dying. And that's what those odds analyses were. I'm not sure if that's what you're asking, but I think that's what we did.
All right, thank you.
You bet.
Okay, sorry about that. Struggling with my my mute there. We'll move over to the Q&A box. Question from Carino. What is the proportion of hatchery steelhead that are from integrated hatchery programs versus segregated hatchery programs? Is there a way to determine catchability differences between the two strategies?
I don't know how you would get to that in the Deschutes. Possibly in another basin where there are both of those types of programs. At least in Oregon, most of the sort of segregated program, or at least most of the integrated programs are more for winter steelhead, and these are all summer steelhead. So most of the summer steelhead programs in the mid and upper Columbia are segregated. So I would assume that most of the fish that are in the Deschutes fishery are from segregated programs. I'm not aware of any integrated programs. That doesn't mean that they don't exist, but at least in Oregon, I don't think there's any integrated programs, possibly in Washington or Idaho, but mostly they're segregated. But we don't really, you could possibly look at the coded wire tags from the creel and the harvest and look at the breakdown of those hatcheries and determine by hatchery stock, but that would be pretty challenging to do, at least for the Deschutes.
Thanks, Jason. So then back to Dr. Hinch, we have a question from Teresa Ryan, and she is asking, in the net injuries, did you test different materials? So, for example, nylon versus cotton.
All of the nets were your classic fish-friendly rubberized versions. So they're the ones that you would buy and be told that they were friendly for landing fish. So they were not, we used basically the same type, different models, but the same sort of material. The material that we used in the sling was sort of more of a standard slick tarp sort of material, which wasn't, it was kind of rubberized, but it wasn't. So we used just that standard, and I showed you a picture of the fin that that was breaking through. You could see how the rubberized material. That was the type of net we used each time, Teresa.
Great. And then Dr. Morishima has another question for Jason. So Gary, do you have that?
Sure. Jason, you mentioned during your talk that you were also looking at Chinook and coho. Do you have any results that would indicate any sort of differential of retention or release of hatchery versus wild coho or Chinook?
Yeah, we don't. So we have an active fishery on fall Chinook in the lower Deschutes, but we don't have hatchery fish. It's all natural production for the lower Deschutes. And coho are recent colonizers of the Deschutes. We don't think they historically occurred there, but they have colonized the basin. But again, that's all natural production, so we're not able to do that, at least in the Deschutes.
All right. Well, thank you. I think that for much of the modeling work and analysis work that's done in terms of fishery planning, the encounters are based upon abundance forecasts of hatchery versus wild fish. And if in fact there's a differential encounter, we're not accounting for that in terms of our modeling of Chinook and coho. So that was where the source of the question was from. Thank you.
Yeah.
Okay, thanks, Jason. Next question is for Dr. Hinch from NOAA. He says hello, and he says he can't recall, but is wondering if any analysis was conducted to compare hatchery versus wild mortalities, and if so, was there any difference for coho or Chinook?
Yeah, I think we looked at that in Chinook, and we didn't see any clear differences. The other interesting thing, though, with Chinook is that a lot of the fish that were hatchery were not marked. I think there was only maybe 15-20% of the fish we captured were marked. It was we did have our genetic work done after the season was over to find out it was about 50% of those fish were actually hatchery. And then when we looked at those fish versus the others, first of all, it's starting to reduce our sample sizes to the point where we aren't going to be easily able to see a difference. We didn't see a difference. So is it a sample size issue or not a sample size? I don't think there was any difference in terms of, whether a fish was getting injured in a certain way, and then thus whether the survival affected them in a different way. But what was surprising was just how much more hatchery Chinook was out there than we originally thought. For coho, many of them, especially the Puget Sound fish, were hatchery fish. We had a small number that were interior Fraser River, and those are quite obviously different. They're much bigger, but I think for a large number of our fish, they were hatchery fish when we came to coho. And I see there's a question from Teresa still there asking about the genomics. Teresa, some of it is already published in that one report that I mentioned, our BC Shrift Fund report, primarily the infectious agent aspect of the genomic works we did. And in terms of the broader panels that we're looking at, I just got to get my grad students to finish. So, we're getting there.
Thanks, Scott. So I have a question. It's mainly, focused at Jason, but you're both welcome to answer it. So just a reminder that this seminar series is, about fisheries management, but it's also about environmental change, right? And so this year in particular, we are seeing some really disturbing early indicators about 2026. So we have drought conditions across most of the Pacific Northwest. Snowpack is less than 50% of normal in many cases, and we have a super El Nino predicted to start this spring and continue into summer. So, I mean, you've both been doing this a long time. Jason, 20 years, Scott, just a long, decades of so me as well. But I'm just curious if how sort of environmental stressors may exacerbate some of the stressors or injuries you've been seeing. If they're, if you're keeping an eye out for the impacts of 2026, how that environmental variability plays out in marine versus freshwater. So, maybe Jason, you can answer first, and then we'll hand it over to Scott.
Sure. I mean, I guess as an agency, we are having open conversations about that question, not just in the Deschutes, but statewide. And I think a lot of basins are very concerned about that. Um, you know, Susceptibility, in low water and, if they're exposed to higher water temperatures, does that exacerbate catch and release mortality? All of those things are certainly being considered, and we are taking it sort of more of a case by case, basin by basin. Particularly in the Deschutes, we're lucky in that we, as I described, we have these base summer flows of 4,000 CFS, and it's very stable. I mean, recently we have seen, like, even now, if you go and look at flows, like, I'm not sure that I have seen flows so low, at this point in the year. And but we're talking, we still have 3,700 CFS, and it's probably not going to go a lot below that. So we are lucky. Yeah, the water temperatures generally, it's a lot of it in the Deschutes is driven by what the ambient air temperatures and how much sun is on the, what the weather conditions are. So we have to sort of play it by ear, but we are lucky in that we have very stable flows and don't have to be as concerned about, flow volume and water temperature as a lot of other basins. But it's certainly a concern, like in the John Day Basin, the Rogue Basin. I mean, they're already in very low water conditions and high temperatures.
And then, Scott, did you have anything to add to that?
Yeah, I mean, it's probably one of the largest issues we're going to have to contend with all of our release fisheries moving forward. Certainly in freshwater, we're already seeing that. In the Fraser River, where we've been studying release mortality now for some time, temperature pushes them over the edge really quickly when you handle and release a fish. And especially if you're creating any injury. But the other issue that is not necessarily injury related, it has more to do with their aerobic capacity and their ability to continue to swim. So when you make them struggle and possibly even injure them a bit, and then you release them, you can often push them into a state of acidosis and anaerobiosis that they can't recover from, and they will die of heart attacks. And we see this when temperatures get above 19 degrees and you start catching and releasing fish, either in purpose or because they're escaping from gear. So I know a lot of jurisdictions stop fishing at 18 degrees. In certain Atlantic Canada, they do. We probably should be doing that too. But we do allow fisheries in the river when they're above 18, 19 degrees, but I don't think many of the fish that get either released from gear or escape from gear are going to survive under those temperatures when the temperatures are consistently above those levels for the remainder of the migration, which could be weeks. We're also seeing that in the marine environment. As I mentioned in my talk, we had a period when we had 18, 19, 20 degrees sea surface temperatures. So although those temperatures are brief and ephemeral because the fish can get into cooler water if they need to, what's happening is that there's disease coming along. So we identified Tenacibaculum for the first time, which in Pacific salmon. And that we just had a paper coming out today or tomorrow on that. And so it's associated with Atlantic salmon farming. And this pathogen, when you interact it with temperature and handling, created lesions immediately on our fish. And we thought we were doing something wrong when we were holding on to these fish, and they started to die within a couple of days. But it just happened to be a bad time for us because the water was really warm and this pathogen was very virulent. But when the temperatures are cooler, we didn't have that same problem. So I predict that we're going to see more disease outbreaks associated with release fisheries moving forward. So I think we're going to have to be really careful about where we have those and when we open them, because water temperature is really important in freshwater for those sorts of decisions. It's going to have to also be in coastal waters.
Thank you for that. And Gary, you have questions?
Yes, just another quick question. Scott, could you comment on any observations from your research on differences in mortality rates between males and females, and what the implications might be for the reproductive success of the population?
Oh, that's a whole nother 40-minute seminar that I used to give.
Okay.
And I'm happy to do it, but I'll try to pare it down to 1 minute. Yeah, we've looked at that quite extensively, and certainly in freshwater during the homing migration, females die at twice the rate as males when you put on any pressures on them, whether it be handling, temperature, high flows. And we've been studying, we have lots of years of data that show that. The mechanisms are quite varied, but have a lot to do with the maturation process and the fact that females are putting a lot of their cardiovascular effort into developing eggs and not to being able to recover from strenuous exercise. And so we call it the greedy gonad hypothesis, because so much oxygen and energy is going into those activities, and so there's less metabolic scope available for females to swim. And so and they're also more susceptible to pathogens. They have higher baseline cortisol, which makes them more immunosuppressive. So in freshwater, this is now well established for females. We just discovered this happens in coastal fish as well when they get really close to their maturation phase. So the last study I showed you, the holding study dealing with coho, we looked at maturation status of those fish over the course of the summer, and the last group of fish looked the same externally, but had higher GSIs and higher baseline reproductive hormone levels. And those fish, the more mature ones, died at a higher rate than the less mature ones. So maturity overlays some of these things. It's probably less of an issue the further away they are from freshwater, but once that maturation clock is ticking as they're getting towards freshwater and they're in the river, then sex plays a large role.
Thanks, Dr. Hinch. We'll go back to the Q&A. We have a question from Jeremy Maynard with regards to coho colonization of the Deschutes. When did this start, and are there any ideas on why?
Sure, Jeremy. I don't really have a why. I do know when so as I mentioned in my talk, we've had the shears trap operating since 1977. Never caught a coho in the shears trap until I think around 95. We maybe got one. And then in about 2005, we started getting a handful, and then it started to build through, 2010, where, we were getting, um, 40 or 50. And, within the last 5 years, now we're getting several hundred just, in our sampling at Shears Falls. And so I think in 2019 was the first year we had a sample size large enough to estimate abundance above shears. And, now we're in the several thousand coho above shears. And, when PGE, Portland General Electric, was preparing for the relicensing of Pelton Round Butte, they did a deep dive, and there was actually a book written about historical salmon and steelhead in the Deschutes Basin, and there was no evidence that they could dig up of coho, historically in the Deschutes, but, we don't know for sure if that was the case. but they did colonize, they have colonized. The majority of the fish that are passing were pit tagging and recovering, or at least through pit tag detections, that about 60% or greater of the fish that we sample are going into the Warm Springs River. And so it's mostly tributary production, but yeah, it's been a pretty meteoric recolonization or colonization, whatever you want to call it, of coho.
Thanks, Jason. So it looks like we have one more hand up from one of our PSC Seminar Series committee members, and that's Diego. So Diego, we're going to unmute you if you want to ask your question. Are you there, Diego?
Sorry, Marisa, this was a keyboard accident. I didn't have a question, but I had followed the questions and they've been very informing, particularly the last one regarding differential mortality on females and males. And I can see direct applications for management.
Absolutely. Thank you, Diego. Well, that is all of the questions that have been posed to the speakers this afternoon. The Q&A is closed. So again, I want to extend our gratitude to both Jason and Scott for excellent presentations, really thought-provoking. We will post these presentations on the PSC YouTube channel in the following days. Thank you for that. And then also, I had kind of alluded to the fact that we may be going into some environmental uncertainty in the months ahead, especially as we begin prosecuting some of these fisheries in the summer months. So the plan for the PSC Seminar Steering Committee is our next talk is going to be in the summer, talking about some of the in-season impacts of the Super El Nino on fisheries prosecuted in the U.S. and Canada. So no date yet. We're still identifying candidate speakers. So if you have any ideas, please reach out to us. But that is our plan for our next seminar that will happen in the summer. And in the meantime, again, thanks for all joining us, and thanks to the speakers. And we will see you next time. So take care. Bye.
Thank you.